Hey guys, I'm glad you're able to find these notes. This is the third lesson in biology for Science 9. Today we're going to be talking about meiosis. But before we can actually get into meiosis, I want to briefly talk about sexual reproduction, because we've talked about asexual reproduction, but we haven't touched on sexual reproduction. And this is really important because meiosis is what gives us the cells we need for sexual reproduction. So, let's look at sexual reproduction versus asexual reproduction. How about the number of parents? Well, for sexual reproduction, it requires 2 parents, whereas for asexual, it only requires 1. And because of that, the offspring for asexual, remember, are identical. They're little clones. Whereas for sexual reproduction, think of yourself, think of your siblings if you have them. You are not clones of your parents. You might be very similar to one of your parents or the other, or both, but you're not identical to them. So these give you what we call genetically different offspring. Because sexual reproduction requires 2 parents, and because of it requiring the combination of 2 cells to create a new organism, this actually takes quite a long time. This is relative for humans. Remember, for us, it takes about 40 weeks for us to have a baby. For elephants, it's almost 2 years. So depending on what type of animal, it's very subjective to how long it takes. But for asexual, this is really fast. Some bacteria can reproduce in 20 minutes, which means that every 20 minutes, the number of those bacteria can double under ideal conditions. So also the cost. Asexual reproduction isn't very costly. Yes, they have to grow before they can divide, but it's not very costly, especially when you think of sexual reproduction. This is very costly. And this is because you need to find a mate, and a lot of finding a mate generally requires some mating rituals. So peacocks with their feathers, a lot of birds with their feathers, it takes a lot of their energy to grow the different colors or the large feathers that they show off. Penguins with collecting rocks for certain breeds. Most animals have some sort of mating ritual that they have to perform to win themselves a mate. So not only does it take you a lot of energy, but it may take a lot of time, especially if you are a type of organism that's not near or very close to others of your species. Okay, so for sexual reproduction, we require 2 different cells to come into one, and these are called gametes. And these gametes, which are also called sex cells, contain half the DNA of the organism. So in humans, this is going to be our sperm and our egg. Both of these are gametes. And we call them 2N. We call them N, whereas a regular body cell is 2N because we have 2 copies of every chromosome. These only have 1, so they're called N or haploid. body cells, which are normal cells, are 2N, and these are diploid. Okay, so now that I've gone through this part, I'm going to go into a bit of a PowerPoint. And the reason why I'm doing a bit of a PowerPoint for this one is because look at these pictures. You guys know I'm no artist. This is way better than I could draw, and I really felt that I didn't do mitosis justice. But yes, you are looking at, what, 9 different stages? And these 9 stages are the ones that make up meiosis. If you look, it's split into 2 different divisions, the first cell division and the second cell division. And if you look closely, these look very familiar because they're very similar to mitosis. Okay, so reproductive cells are formed by a process called meiosis. We have 46 chromosomes, 23 pairs, and like I said, we have duplicates of all of our chromosomes. So we have 23 pairs. Each of these pairs are called homologous chromosomes, and during fertilization, each parent contributes 1 chromosome of each pair. So each parent contributes 23 chromosomes. There are times where this goes astray, and you may give more than 23 or less than 23, but this generally ends up resulting in some sort of syndrome or disorder, depending. A common one for this is Down syndrome, where they actually are given a 24th chromosome, usually from the mother, but not always. So homologous chromosomes are really interesting because they are the same size and the same shape, and they carry the same genes. But they may not be the exact same version of that gene. Different versions of genes are called alleles. And yes, it's a really weird name, alleles. And these are just different versions of that gene. So some of these are like eye color. You may have an allele for brown, blue, emerald, or gray eyes. Hair color, blonde, red, brown, and black. So this is why your parents may be blonde and brown hair, and you end up only being blonde. Even though you may have a blonde and a brown hair gene, one may show up more than the other, and we'll get into that in the next set of notes. So gametes or sex cells, remember these are our sex cells, are haploid. So that means they have half the number of normal chromosomes. When they combine together, so when you take a haploid sperm and combine it with a haploid egg, this is, remember, N, this is N. This will give you a diploid zygote. Every single organism that reproduces using sexual reproduction starts as a zygote. It's the first cell. So you have N plus N will give you 2N. This will have the full complement. And like I said, body cells, all body cells are diploid. So skin cells, liver cells, they're all diploid. So what you'll have is you'll have this, the male gamete, the female gamete. They will combine together. They'll give you the zygote. That zygote will divide, become an entire organism, or stay as one, depending. But it will grow into an adult or male. It will undergo our meiosis right here, and it will develop one or the other sex cell. Okay, so cells that produce gametes undergo a special type of cell division called meiosis. And meiosis is when a diploid cell divides twice to produce 4 haploid cells. This allows offspring to be genetically different from the parents and from one another. If you think, again, if you have siblings or your friends have siblings, they're not identical. There might be similarities, but none of them are identical. And this is because we have some crossover. So if you look, we have in interphase our chromosomes. During interphase, they duplicate. You have your sister chromatids. And then when they go into metaphase, they'll actually cross over and they'll switch parts. And this isn't intentional. This is basically you're jamming a whole bunch of chromosomes together, and some of them accidentally cross over and squish together. So as you can see, we have some blue on the red and some red on the blue, and it splits the first time. And this will split the homologous chromosomes into two different cells. So we have one of this chromosome, one of this chromosome, one of this one, and the other one of that one. Once we've done that, it'll undergo meiosis 2, and that's when we're actually going to rip the sister chromatids apart to give us our chromosomes again. And if you look on this, it shows you that none of these four cells are exactly the same. Big red with a little blue with an all little blue. There's no other that's just little blue. There's no other that's mostly red with little blue, and so on and so forth. So all four of these cells are different from the others. Okay, so it'll undergo interphase just like before, but there is no difference between interphase for mitosis and interphase for meiosis. It's going to duplicate its organelles, it's going to grow in size, it's going to duplicate the chromosomes, it's going to do everything it did before. It gets different when we enter prophase 1. So in prophase 1, again, still very similar. We're going to have the nuclear membrane that begins to disappear. Our DNA is going to condense into our duplicated chromosomes, or our sister chromatids, and our homologous pairs, this is what's different. Our homologous chromosomes are going to be paired. So this picture is from your textbook. This one shows it a bit better. You can see that these two big X's are paired up. These two little X's are paired up. And that's really important to see. We're going to have our spindle formation with our centrioles. But really this is the key difference. So once we pass through our prophase, we're going to go into metaphase. Metaphase, the spindle fibers guide the movement just like before. The homologous chromosomes pair up in the middle. So instead, in mitosis, we had one single file line. In metaphase 1, in meiosis 1, we actually have pairs that line up in the middle. This is where the crossing over happens, because as you can see, they're really squished together. This is much prettier than what it actually looks like. Even this is pretty considering what it really looks like. Again, this one's from your textbook, and it's very neat. If you look at a video on YouTube or a picture from Google, you'll see that this is actually a huge mess. It's just a mass of lines down the middle. And this is when crossing over occurs. So when you have them and they're mixed up, you'll have them where they cross over each other. And when they cross over each other, they're actually going to unattach from themselves and attach to the other one. And you're going to have a mixture. So this is why some of your genes, they're not all your dad's genes, they're a mix of your mom and your dad's genes, because you're getting a mix just in one chromosome alone. It's also what allows us to have such variety, because you're not just going to get a straight blue one. You're going to probably get blue with some red, and depending on how the crossing over occurred, it could be more or less red. So once metaphase is done, we go into anaphase. And the only difference is, now that the homologous chromosome pairs separate instead of the sister chromatids. So if you see, we have the X's being pulled apart instead of the single lines. And then we have telophase 1. And telophase 1 are two haploid nuclei are formed. Each nucleus contains half of the original because we have one big red, one little blue, one big blue, one little red. The cell divides and two cells are formed. So we can call this cytokinesis, but you may also hear this be called interkinesis. And the reason why it's called interkinesis is because it's in between meiosis 1 and meiosis 2. So prophase 2. This kind of skips, there's no interphase in between the interkinesis and the prophase 2. It kind of goes straight into it. And this is where you're going to have your nuclear membrane disappear again. Your DNA already exists as your chromosomes. But now, instead of like in prophase 1, these guys are not diploid, they're haploid, and they're all unique. So as you can see, in prophase 1, your homologous chromosomes paired up, and your spindle was formed. Your spindle is still formed, but there are no homologous chromosomes to pair up. There's just single chromosomes in there. Then in metaphase, we get back to our single file. There's no chromosome pairs, they just line up just like in mitosis. So remember in metaphase 1, we had our homologous pairs pair up, so we had two X's. It was not single file, but two pairs in line. And then in metaphase 2, we have two separate cells, which is why we always show them, but they're single file again. Anaphase, the copies of DNA or the sister chromatids are separated. This is when we call them chromosomes again. So in anaphase 1, we had the full X's or the full homologous chromosomes being pulled to the poles. In anaphase 2, we just have the sister chromatids which are being pulled apart. So this middle is a centromere, and this is where the two attach. And then we have telophase 2, and in telophase 2, we have 4 nuclei which are formed because we're going to create 4 cells. And then the cells begin to divide, and they undergo cytokinesis, and they form our 4 cells. So remember in telophase 1, this one was pretty similar. We had our nuclear membrane reform, we had our cells start to cleave, which means separate into 2, and that's about it. And then in telophase 2, it's very, very similar. The only difference is that we have 4 cells instead of 2. Okay, so going over this again, this first one where we have them condensed into chromosomes, we have our spindle fiber forming. This is called prophase 1. And yes, you do have to put the 1 because we have to distinguish between prophase 1, prophase 2, and prophase from mitosis. So it's very important to put the number or the lack of number depending on the situation. Okay, when they meet in the middle, that is going to be, well, and we have our X's meeting in the middle, not single file. So this is our metaphase 1. We have our X's being pulled apart, not just our sister chromatids being pulled apart. So this is our anaphase 1. And then in this last picture of the first cell division, we still have X's in our nucleus even as it's reforming, Because we still have X's, I know that the sister chromatids haven't been separated, so that tells me it has to be telophase 1. So once that has occurred, we move on to our second cell division. So we still have X's, but we only have one of each chromosome. Our spindle is forming. That means this has to be prophase 2. And I don't require you to write it on the top and the bottom. These cells are all in sync in this picture, which means whether you write on the top or the bottom, I refer to that for both of them. You can also write in the middle, but with your assignment today, you're actually going to have to do descriptions as well, and you probably won't have enough room in here. Okay, so we have single file line, but we only have one of each chromosome, which means this has to be metaphase 2. We have our sister chromatids being pulled apart, but again, we only have one of each, which means this has to be anaphase 2. And we have our nuclear membranes reforming, but we only have one copy of each chromosome, which tells me this is telophase 2. And this last one is cytokinesis, because this is where the nuclei have already been formed here, but we're just separating the cytoplasm over here. So it gives us cytokinesis. There is one phase that was missed, actually 2 phases that were missed. We were missing our interkinesis, and in this first part, we were actually missing our interphase. We didn't have a picture of it. In your assignment, there is a picture for interphase, so you have to be really careful about that. Okay, well, your assignment today is the meiosis and mitosis assignment. And there's 2 ways to do this depending on whether you have a printer or not. If you have a printer, I have 2 documents in my Google Drive. One that says meiosis assignment cards. And one that's just meiosis and mitosis assignment. So this one is the one that has the directions. This is what you have to follow. And then what you need to do is, if you have a printer, you need to print these out. If you don't have a printer, you need to draw them. Beware, because there are letters beside each of the pictures, A, B, C, D, and I need to see those letters with your drawings if you've done that. And what you're going to do is, these cards contain both mitosis and meiosis. And yes, they are random. Not only are they not in order, but meiosis and mitosis are mixed together. And what I'd like you to do is, I would like you to 1, separate them into mitosis and meiosis. 2, put them in order from interphase all the way through to the final cytokinesis. And then you're going to have to color them if you have pencil crayons or felts or whatever you have to possibly color them with. And then you need to do descriptions and answer some questions. So this is going to take us a little bit longer. I am not going to assign anything else until Friday, and Friday I'm just going to give you the quiz. And the quiz is going to be short. It's going to be like 15, 16 questions. Most of them multiple choice, maybe 1 written, and that's it. Just because there's one question I really want to ask that I haven't had a chance to yet. The quiz on Friday is going to be all about asexual reproduction. So that video, those notes, the additional video about the tree of 40 fruit, but that's about it. So if you have any questions, please let me know. If you've already attempted this assignment, please go back and look at it and make sure that you've hit all the requirements, because I do have my marking scheme on the directions page. And I will, like always, accept another copy if you think you can improve what you've done before. Beyond that, I hope to find you guys safe and healthy, and you'll hear from me later this week.